Welcome to today's cutting edge conversation titled Addressing COVID-19 During Uncertainty, sponsored by Integral Molecular and OM1. Thanks so much for joining us today. So today our guest speakers are Dr. Hayley Crawford from Integral Molecular and Dr. Carl Marcy from OM1, who will discuss how Integral Molecular and OM1 are contributing to COVID-19 research. Just a few notes on background of COVID-19 research and a few of the challenges associated with it. So we'll start with a few definitions to get us all on the same page here. So coronaviruses are actually a family of viruses, which in humans mainly cause upper respiratory illness. There's 4 of them that cause mild cold-like symptoms. Another coronavirus, which is a big deal for about 8 months after it emerged in 2002. You might remember SARS. And yet another one is responsible for the recent global pandemic, which has killed many, infected more, and impacted really everyone. It's impossible to understate how influential the disease really is in so many ways. And on a personal note, I actually got COVID just the week before last. I'm triple vaxxed and still had a high fever for 3 days and a persistent cough, which I haven't managed to shake quite yet. But that's just me. So brief timeline here. So mRNA was discovered in the early 1960s, and research into how mRNA could be really delivered into cells was developed in the 70s, which has sort of led to the vaccines that recently came out. So this research continued for many decades. In 2002, we saw the first strain of SARS-CoV, and then it came about again in late 2019 with the vaccine coming along in 2021. So despite an unprecedented international push to really quickly understand how the virus works and to take action to mitigate it, there's still a lot we don't know about it. But it's not from a lack of effort. In an ultimate trial by fire, researchers, healthcare providers, policymakers around the world really were tasked with finding solutions, and each one with its own unique set of challenges. I mean, for example, there's a lot that we've done already. Data scientists and epidemiologists develop systems to track the virus, how it's evolving, assess risk. The necessity for safe and effective vaccines led to, an immediate outpouring of research from pharmaceutical companies, priority treatment from regulatory agencies, stepping up of production, vaccine production facilities, and really massive distribution programs. It usually takes 10 to 15 years to develop a vaccine. But some countries were already vaccinating select groups as early as December 2020, which is just 12 months after the virus was identified, which is pretty amazing if you think about it. There's also been a lot of research done to understand what's actually effective at lessening risk of infection and severity, leading to the stuff like lockdowns and mask mandates. Reminders to practice social distancing in these uncertain times. There's also been public health officials who've drafted up guidelines and made decisions on how to enforce those measures. And there's also been, of course, a tremendous amount of ongoing work dedicated to finding new treatments with the goal of inhibiting viral entry into cells or replicating, or to curb the hyperinflammation associated with it. There's also been, chemists and immunologists developed, PCR and protein-based tests. Some of those things are pretty cool. The week before last, when I had just a slight cough at that time, my partner grabbed a box of rapid tests from the grocery store for free. I went home to my kitchen, swabbed my nose, mixed it with a buffer, dropped it in a little cartridge, and 15 minutes later, confirmed the reason why, I feel like complete garbage for the next 7 days. Some ongoing challenges that have been done so far, or that are still, ongoing. There's the fact that the impact of COVID-19 can vary tremendously based on where you are. So different countries or even different states have dramatic differences between them in terms of morbidity and mortality. And yeah, things like access to health care, vaccination rates, environmental conditions, public safety measures, and compliance, those all have a role. But there's also other factors in play. Different regions actually study the virus differently, and that makes a difference too. So different surveillance tactics, different means of tracking the virus, different reporting methods, differences in even data quality and accuracy, testing capabilities and methodologies. We're looking at different data sets between different places. And compounding on this issue, individuals, whether consciously or not, will misuse and misinterpret the data. For example, in a recent article, Lambert et al. Compared demographics, public health, economy, policy, and environment to test their association with mortality. And they came up with a conclusion that high-income countries were hit the hardest, and that the burden of COVID-19 was not affected by more stringent public health decisions. But what Lambert failed to take into account was the fact that these countries with higher incomes also tended to be older. And of course, the risk of severe COVID-19 illness is much greater in older individuals. And also excluded from that analysis was the timing and compliance of public health measures. It's been shown that mortality is reduced in countries that initiated a strong early response versus one much later along the line. And so, that's just one example, but you can see how, having a article that says something like public health measures don't affect mortality, you can see how that would add fuel to the fire for, policymakers, conspiracy theorists even. On a more human note, another challenge is that, COVID-19 has been really lousy for social interaction. The pandemic's been a massively destabilizing force for all kinds of our human institutions. We've changed the way we've lived our lives, and the way we do business too, of course. Working from home is more common than ever, and in many cases, we've traded in-person meetings for virtual ones, which in some cases, is convenient. But virtual environments just can't replace the human interactions that we have in person. The pandemic's also had a huge, specifically social impact. Even those who have never gotten sick, it's undeniable that the social isolation and uncertainty has psychological consequences, resulting in increased stress and anxiety. I mean, personally, like I see my friends and family less often, and maybe some of you feel the same way. And with that, I'm glad that I could end my section on such a happy note. But with that, I'm looking forward to hearing from the presenters. And with that, I'm pleased to welcome Dr. Hayley Crawford. Hayley, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you so much, Liam. Good morning. I'm Hayley Crawford from Integral Molecular, where I'm the virology project leader. I'm so happy to have been asked to take part in this panel about uncertainty during the COVID-19 pandemic, and I'm looking forward to hearing Dr. Marcy talk after me about the psychological implications. So I'm going to be coming to you from a purely lab-based scientific point of view. We have been in the lab since the beginning of the pandemic, when things were very uncertain, and arguably they still are. With each emerging variant since the beginning of the pandemic, our team has sought to keep abreast with developments and adapt along the way, knowing that myself and my team were responding in real time to a public health emergency has been one of the most rewarding parts of my career thus far. And today I'm looking forward to sharing with you some of the research tools we've developed and how they might support your team's virology research. So, Integral Molecular is a biotechnology company founded by virologists in Philadelphia, where I'm based. Our original technology was lipoparticles, which are enveloped virus-like particles used for the display of membrane proteins. And we also make reporter virus particles, which I'm going to be talking about today. Sorry, my slides are skipping. Here at Integral, we work on numerous viruses listed here. And since the beginning of the SARS-CoV-2 pandemic, we have added not only that virus and all of its variants to our catalog, but also SARS-1 and MERS. We have great relationships with hundreds of academic, industry, and pharmaceutical labs, and we have multiple technology platforms and products that are used all over the world. We publish papers on our own research, shown here, and we also collaborate on publications and patents with our partners. So to give you the lowdown on RVPs, our reporter virus particles are fusion-competent pseudoviruses that are capable of one round of infection of target cells, and so they're safe in a BSL-2 lab. Safety is the most crucial aspect of working with viruses, and our RVPs are not only safe, they are antigenically identical to live virus, high throughput, and are easy to use. Inside the core of the RVP is a reporter gene that is expressed inside target cells after membrane fusion, and this enables the detection of infected cells. The RVPs have been optimized to overexpress the membrane protein of interest on their surface, in this case, the SARS-CoV-2 spike protein, and they contain no other genes from the virus of interest. And we also have very strict QC criteria that these reagents must pass before we release them. Our RVPs can be utilized in infectivity assays and in serum and antibody neutralization assays. We have two types of readout for these assays, either green fluorescent protein or GFP, which is detected via flow or microscopy, as seen in this photo on the right. We also have RVPs that contain Renilla luciferase, and following infection, relative light units can be detected using a luminescence plate reader or a luminometer. We have VSV RVPs available as a negative control. Sorry, my slides keep skipping. And we have neutralizing antibodies against SARS-CoV-2 as well, which I will talk about a little bit later. With your first order, we will send out a monoclonal HEC293 T cell line that stably expresses ACE2. And all of the protocols you will need are available on our website too. If you haven't downloaded them for a while, make sure you get them today, the latest versions. And in addition, we have a team of PhD scientists that are able to troubleshoot any issues you may have. So our SARS-2 RVPs have been shown to be comparable to the historical standard plaque reduction neutralization test, or PRINT, by our customers at the University of Pittsburgh, among others, shown here on the left. The results you will achieve with our RVPs will be accurate, reliable, and reproducible, no matter which lot you use or what volume you input. Viruses are constantly changing, and new variants evolve and spread, as clearly demonstrated by the COVID pandemic. This chronological phylogenetic tree shows the increasing number of mutations that the virus is accumulating on the y-axis versus time on the x-axis. Delta is in turquoise, and 2 Omicron variants are in shades of orange. The frequency figure below shows which variants have predominated over the past year. The world is now facing mainly Omicron and its sublineages, shown in the oranges, and the virus is still evolving. To be able to study these constantly emerging variants, we have utilized our RVP technology to generate every variant that the World Health Organization declares a variant of interest or concern, as well as other variants that our customers and collaborators are interested in. As of today, we have 73 SARS-CoV-2 variant RVPs in our catalog in both GFP and luciferase reporters, and we have also produced SARS-1 and MERS-2. We also have an alanine scan library of SARS RVPs, and we take custom requests. In addition, our catalog contains dengue, Zika, chikungunya, and VSV RVPs, and we're adding more viruses soon. This huge variety in our RVP catalog enables neutralization profiling for monoclonal antibodies or immune responses in patient or vaccinee sera. For example, on the left, our reference antibody neutralizes the D614G variant, as well as alpha, beta, and gamma variants in shades of blue. In the middle panel, we tested a monoclonal antibody from a customer, and while it neutralized the D614G variant, it poorly neutralized the beta variant. Likewise, on the right, sera from a collaborator neutralized the D614G variant well, but not the other 3 variants. This profiling is essential for insights into vaccine responses and therapeutic antibody efficacy. We also have exciting new additions to our catalog. We recently discovered a neutralizing antibody that neutralizes all the current circulating Omicron strains on the left. And we also have a neutralizing antibody that strongly neutralizes the Delta variant. These can be used as positive controls in your neutralization assays, and you can get more information on these on our website. If you're preparing a vaccine or a therapeutic antibody for licensing, our RVPs check all the boxes. The FDA recommendations to vaccine developers state that they should assess the immunogenicity of modified vaccines against variants, and that neutralization assays with wild-type SARS-CoV-2 virus or pseudoviruses may be used. For developers of therapeutic antibodies, the FDA recommends that they implement a potency assay to confirm consistency and potency of each lot, report the neutralizing activity against variants, and pseudoviruses such as RVPs may be used. RVPs are not our only technology to support antibody development informed by FDA guidances. Our shotgun mutagenesis epitope mapping platform helps to address the FDA recommendation that antibody developers should thoroughly characterize epitopes to identify escape mutations in circulating variants. Our shotgun mutagenesis platform rapidly maps antibody epitopes at single amino acid resolution, and for SARS-CoV-2 antibodies, we will give you results within 4 weeks. Our membrane proteome array can help address the FDA recommendation that preclinical studies should evaluate potential off-target binding of antibodies. Our MPA screens antibodies for off-target binding against 6,000 human membrane proteins, and we will provide you with an IND-enabling report in approximately 4 weeks. So lastly, this slide shows data from our customers at Immunome who used our RVPs as well as our epitope mapping and membrane proteome array platforms for preclinical profiling of their 3-antibody cocktail against SARS-CoV-2. On the left, they used 10 of our RVP variants, and this showed that their antibody cocktail neutralized these circulating variants from alpha through mu. We were able to map the residues in the epitopes for each antibody, and we found that they have unique and cryptic or hidden epitopes. And our membrane proteome array platform proved that their antibodies bound only to the target of interest and did not have any off-target specificity. Sorry for the jumping around. Immunome subsequently submitted an IND application to the FDA for this potential therapeutic. You are able to purchase all of these reagents, the RVPs, cells, antibodies, through our website, but also on scientist.com. So look for us on scientist.com. Thank you so much for your attention today. And thank you to my amazing team at Integral Molecular. We are very proud of how our technologies have enabled scientists around the world to progress in the fight against SARS-CoV-2. Our team continues to adapt to this changing pandemic, and we look forward to closely working with you to ensure you succeed in your virology research. Thank you.
Shifting gears just a little bit, I'd like to welcome our next speaker, Dr. Carl Marcy. Carl, welcome. The floor is yours whenever you're ready.
Great. Thanks, Liam. And thanks to Dr. Crawford for a terrific presentation and obviously very important work. We're now going to shift gears, I would say, significantly. And we're going to move from the world that is microscopic of virology to the more macroscopic world of big data and mental health. So my goal today is to talk to you about a number of topics, but first to introduce, let's see if we can get this to work. From my perspective and from an OM1 perspective, how real-world data can inform our understanding of some of the uncertainties around COVID-19. And we're going to talk a little bit about some new data that we haven't released that you will be seeing first here today, and then my sort of interpretation and understanding of what that data means. But again, my name is Dr. Carl Marcy. I describe myself as a physician, scientist, and entrepreneur. As a physician, I'm board certified in psychiatry and have maintained a small clinical practice through Mass General Brigham in Boston for the last 20 years, where I predominantly see outpatient psychiatry, psychopharmacology, and therapy, which is really mostly about depression, anxiety, bipolar disorder, and PTSD. As a scientist, I spent the early part of my career trying to understand the neurobiology of empathy. I have a number of peer-reviewed papers and 7 patents, and a shameless plug for my upcoming book called Rewired, which is looking at the impact of mobile media, information, and communications technology on the developing brain. And as an entrepreneur, I've been involved in 4 different companies at the intersection of healthcare and technology. And what excites me today is working with OM1, where I joined several months ago because we had an opportunity with a new data asset, which I'll describe in a moment, and then we'll talk a little bit about COVID. So job 1 is to understand what real-world data is and how it's acquired and processed and used, which will give you a sense for how we generate some of the insights I'll describe around COVID. Why real-world data is important from a mental health perspective. And then again, I'm going to review some new data that we just are about to release that we'll talk about here for the first time on depression, anxiety from a real-world data perspective. And then we'll jump to some Q&A. So first, a definition. As Liam started with the definition. And this comes from the Food and Drug Administration on what real-world data is. So real-world data are data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. And the key here is a variety of sources. So think of this as big healthcare data, right? So we get data not only from pharmacy and insurance claims, but we also get data directly from electronic health records, from patient exchanges. We get data from labs. We also get data from mortality and other sources. And we combine them in the cloud. And having data isn't enough. You also have to generate insights. And that's where real-world evidence comes in. So real-world evidence is the clinical evidence regarding the usage and potential benefits or risks of a medical product, typically a device or a pharmaceutical, derived from analysis of real-world data. I think an important question is why now, right? Why is real-world data and real-world evidence becoming such an important and hot topic? Well, I think there's 4 trends that are coming together to really meet the moment. And the first is the broad availability of healthcare data, right? We all have the experience of going to our clinicians now and people typing in front of computers and using electronic health records. More and more, we're filling out surveys through patient gateways and apps and other things. So the ubiquity of healthcare data is job 1. Job 2 is having the tools to do something with it, right? So this is where advances in data science and artificial intelligence allow us to do the same thing that Google and Facebook do with consumer data, but make this in service of our health and apply these tools to large data sets that can help clinicians make better decisions and to help patients navigate their lives in better ways, and also to help pharmaceutical companies bring new treatments to market. I think the third trend is we now have a generation of consumers who are used to having everything personalized. The advertising we see is personalized. Our groceries are personalized and delivered to our door. There's convenience everywhere we go. So why shouldn't healthcare be more personalized and more tailored to the individual and the individual's needs? And I think this is where big data can really make a difference. And then the final force, and probably the one that's least familiar to most people, is that you have to have investment. And so in 2016, the federal government passed the 21st Century Cures Act, which, among other things, provided funding to the food and incentives to the Food and Drug Administration to go beyond randomized clinical trials, which are incredibly important, but often do not generalize to the real world. And to incentivize life sciences companies, pharmaceutical companies, and the FDA to embrace real-world evidence so that we can actually see outcomes as they play out in post-market. So I think those are really important. How do we actually process this data? Again, very quickly and at a high level, we acquire data that is relevant. And we do that from a number of sources, as I said earlier. The next important thing is that we have to process that data to research and regulatory grades. And this is really where our tech teams and our clinical informatics groups are working very hard, because big data is also dirty data. It needs to be cleaned up and it needs to be organized in the cloud. And then the final step is to have the tools and the talent to mine it for insights and impact. And at OM1, we have 3 technologies to do that. The first is we call OM1 Origin, where we are connecting to our different platforms and networks and data sources, and we're de-identifying them, and we're tokenizing them, and we're organizing them in the cloud. Step 2, beyond that organization, which is initial, is to process, to enrich variables, to make sure they're where they're supposed to be, to fill any gaps that we can. And then the final step is, again, to apply data science and artificial intelligence and basic biostatistics to generate insights. And the advantage of all that is to have a large data set in the cloud that combines data from multiple sources. So we have patient-related outcomes. We have clinical encounters from EHRs. We have open and closed claims data from insurance companies and pharmacies. We have direct data on types of providers. We have laboratory data, vital signs, mortality data, different data on different types of drugs and devices used, procedures that occurred, social determinants of health, and in some cases we even have consumer data like household income that we know play a role in how folks are doing. And the way we work with clients is threefold. Some of our clients will directly work with us to do analytics and research on our data. Others will work with us to prospectively collect data that might be missing, that's not routinely collected in the real world as a complement. And then the third way we work with clients is direct licensing of our premium data products. All right, so let's talk about mental health. Even before COVID, I think it was very clear, at least to those who gave it thought, that in this country, at least, we were in a mental health crisis. And among the issues related to that crisis were the cost of care. And so I put this up. This is pre-pandemic, 2016. But when you look at the cost of mental health disorders in dollars in this country, you see that it is the number one and the most expensive group of disorders that we have to deal with. And what I always remind people is that mental health makes physical health worse. So people who have arthritis or diabetes or heart disease and depression or anxiety, or in many cases both, will have a harder time getting treatment. And that it's really important for us not to always work in silos and to remember that the patients we're treating with really have mental health, physical health, and social health issues that all need to be addressed. And so we had this growing crisis and real problems that the industry was just starting to respond to. This just shows you the investment, at least again, pre-pandemic 2018, where we see the pipeline, which for many years had gotten a little dry, is now starting to be enriched with novel molecules, with novel mechanisms that can treat everything from Alzheimer's, dementia, to treatment-resistant depression, to post-traumatic stress disorder, ADHD, et cetera. So I think the next 5 to 10 years are going to be very exciting. But I don't think as a society we can wait. And so in order to speed things up, that's where this kind of data comes in. So I joined OM1 at the beginning of this year because we had acquired a massive network of specialty data in the mental health arena. So we've increased our network to include 9,000 mental health clinicians caring for over 3 million patients, followed longitudinally in 2,000 clinics and in all 50 states. It is a massive data set that we are working with our partners in industry to mine for insights, to bring treatments for mental health to market faster. All right, so let's talk a little bit about COVID and what we found. And I think, again, it should go without saying the importance of this virus and how it's touched all of our lives and how many people. I think it would be hard to avoid a discussion or a headline that didn't sort of make a commentary about the mental health aspects of COVID-19. When we're doing things like social distancing and new terms like Zoom fatigue are coming up every day, we have to really sort of zoom out for a second, pardon the pun, and really consider the fact that we have an opportunity, I think, because of COVID-19, to have empathy with people who suffer chronically from mental health disorders. And what do I mean by that? Well, who among us didn't have a moment of sadness or loneliness that every patient with depression has at some point in the last 2 years? Who among us didn't have excessive worry that we couldn't control and a sense of impending doom that patients with anxiety and PTSD sometimes have? And I know, particularly in the early part of the pandemic, when I was asked in a word to describe my experience, and it came out, it was surreal. And almost surreal to the point where I was losing touch with reality, which gives me a little bit of a sense for what patients with real psychosis actually have. So I think we've all now at least had moments of what is the lived experience of patients who are chronically suffering from mental health issues. When I was working with the team and we were thinking about this talk, I thought, well, we know the headlines show that people are more depressed and anxious, and we see that from survey data. And we know that clinicians are overwhelmed, and the wait lists to have adults, let alone children, seen by a mental health profession have gone up and up and up. And we know from other data that utilization, at least as measured by trips to the ER for suicide, particularly in young people, but adults as well, has also gone up. So we would expect, from a real-world data perspective, to see a simple hypothesis, which is that the prevalence of the diagnosis of major depression and anxiety disorders would have also gone up. And so what we did is we took March 2020 as an index date, and we looked 12 months before and 12 months after at the prevalence of anxiety disorders. And we were surprised to see that it didn't change. In fact, if anything, it went down a little bit. And that was a little surprising. What you're looking at here are just the pre-pandemic ratios of patients in our real-world data cloud. Here, we're looking at obviously about 12 million patients. And prior to the pandemic, those 12 months before, you see about 3.6 million patients who have a diagnosis of anxiety. And then post-pandemic, it goes down to about 3. But you also see that the patients who do not have a diagnosis goes down a little bit. And so we're going to come back to that in a second. The next thing we found, looking at which should be the next one, which is, let's see here, sorry. Depression. Those of you paying attention will see that this looks very similar. In fact, the drop is even higher. So how is it that in a pandemic environment, 12 months into it, we're seeing a decrease in the diagnosis? Well, I don't have slides on this, but I will tell you, when you look at utilization, when you look at global utilization from a real-world perspective, whether it's inpatient admissions, ER visits, or outpatient visits for mental health, we see a significant decrease. And what happened for the first 3 to 6 months during that 12-month period of the first year of the pandemic? Everybody stayed inside, and things got shut down. So people weren't seeing clinicians as frequently as they used to, and so the rates of diagnosing something that clearly was happening more frequently went down because we didn't have access to the licensed clinicians to make the diagnosis. But does that mean we were less depressed or less anxious? And the answer to that is clearly no. So here what you're looking at are two what we call patient-reported outcomes. The GAD-7 is a generalized anxiety disorder questionnaire with 7 questions. It's a screening questionnaire. And the PHQ-9 as the patient health questionnaire with 9 questions that screens for and diagnoses depression. And when you look at that same cohort and you look at their scores, you see that the anxiety has gone up significantly, and even depression has gone up, close to 2 points, which isn't a lot on the PHQ-9 scale. But my last slide here on this data, we can get by it. Let's go like this. Takes a closer look at depression. And what we see here is how when you take the categorical perspective of depression, which is the absence of the disorder, mild, moderate, severe, and you look at the pre-pandemic levels, which are in dark blue, and the post-pandemic levels, which are in lime green, we see a significant shift. And the good news is for those who didn't have the diagnosis before, mostly they have been stable. But for those at risk who had a diagnosis and were in treatment, because remember, this is real world data from people going to see mental health specialists, we see an increase in every category. More mild depression, more moderate depression, more moderate severe depression, and more severe depression, which again is consistent with the headlines. And I think one of the takeaways here is that we are dealing with an environment where access to mental health professionals was a problem before the pandemic, and it's gotten worse. And I'll give you one anecdotal piece of information, which is the waiting list in my clinic prior to the pandemic was 3 to 6 months. It's over 2 years right now. So there are people suffering who can't get a diagnosis, who can't actually fill out a PHQ-9 or a GAD-7. And as a society, we need to really think about our resources and how we approach things. So I'll stop there in the interest of time, and we can switch over to Q&A at your leisure then.
Fantastic. Yeah, really interesting presentation, Carl. Thanks a lot for that. I'll add in Hayley back here, and then we'll get right started with the Q&A here. Hayley, actually, I think I'll ask you to lead the first one here. Are the RVPs available through scientist.com?
Yes, they are. They are available. If you go to scientist.com and search integral molecular, you'll find them. And then we also have them on our website, integralmolecular.com.
Awesome. All right, Carl, this one will be for you. So thinking about the depression data, can you talk about your approach to understanding response, remission, and relapse rates?
Sure. Yeah, it's a good question. So if you recall the PHQ-9, which again is a questionnaire that most people should be familiar with. If you go to your primary care doc or see a mental health specialist, you probably have filled it out. We use a commonly accepted academic definition of a change in score of 50% as response. So if I'm at a 20, which is at the edge of severe depression, and I go down to a 10, I've dropped down now to mild depression. That's a significant response. And then remission sort of depends on who you're talking to, and we can adjust our data accordingly. But the strong definition of remission is below 5. And then some people also use below 10, where you've gone from severe to really mild and more manageable depression as a depression response. But the key here is that you have those patient-related outcomes over time so that you can assess change in response to various treatments.
Yeah, great answer. We do have lots more questions, but as a reminder, if you do have a question that you'd like to ask, you could submit it using the Ask a Question panel shaking on the screen there. All right, Hayley, one for you here. Do you have any RVPs available for viruses other than SARS-CoV-2?
Yes, we do. So before the pandemic, we mainly made dengue and Zika RVPs. And we currently have chikungunya, we have VSV, SARS-1, the original SARS, MERS, and then we have numerous viruses in R&D, such as Ebola, Marburg, all other kinds of dangerous and airborne viruses that are safe as RVPs.
Right. Yeah. Perfect. All right. Next one, Carl, for you. Here's a good one. What uses of AI have you had success with in mental health and neuroscience?
Yeah, so I think the one that comes to mind is in treatment-resistant depression, right? So treatment-resistant depression is not classically a diagnosis. It's not even in the DSM, which is the Diagnostic and Statistical Manual for Disorders, the Bible for psychiatry. But it is a known clinical entity, and it has an academic and regulatory definition of the failure of two antidepressants of adequate dose and duration. So I'm diagnosed with depression, and I'm given not one but two medication trials that are considered decent, and I'm still depressed. And while that's a very fine and important definition, it's really hard to do, even with our data, to tease out, well, what is an adequate dose and duration? Are people switching medications because they're having a side effect, or they're just not tolerating it, or are they switching because they're getting better, right? And so in our data, we can tease out some of those details, but it's incredibly hard for most people to actually do that. So one of the things we've done is said, look, what if we can take our data where we can have as a kind of ground truth where, clinicians have literally used the label treatment-resistant depression. We can take that regulatory definition and other things and apply it, and then create a model of those patients and then apply it to other patients as a kind of patient finder, right? And so patient finder is an AI tool that we've developed that allows us to identify people either at risk for going on to develop treatment-resistant depression, or people who have it but don't know it. Now, why does this matter? Well, because you're gonna treat those patients differently. You're gonna treat them more aggressively. And going back to the cost estimates I showed earlier, the thing about treatment-resistant depression is, let's say it's between 10 and 30% of depressed patients have treatment-resistant depression. They spend about half of every mental health dollar. Right, so because they're much harder to treat. So that's one example of how we're using AI.
Yeah, fantastic. That's a great example of a good way to use AI with these gigantic data sets. In the Hayley, in the chat, you said that you have a question for Dr. Marcy.
I do. Dr. Marcy, so in the last slide, your last data slide, you showed that pre and post-pandemic, people without depression, numbers were the same. People with mild through severe, it went up. So does that mean that if you had a form of depression before the pandemic, it got worse, but if you didn't have it before, you didn't get it during? Are you saying that the pandemic didn't bring about depression in the people you studied?
I think that's one interpretation. Now, I'm going to caveat that this is a preliminary analysis, and this surprised me as much as it surprises you. But I think what it highlights is the fact that, and we know this from a mental health perspective, is that some people are just more vulnerable than others. And so, whether you're privileged to be able to work from home and we're able to keep your job, and to have the financial resources to provide, food and shelter, during the last 2 years, I think really separates and protects people from that social isolation, some of the fear and anxiety that presents as symptoms, right? So if you ask people a survey of just, do you feel more anxious? Most people are like, hell yeah, I'm more anxious, right? Do you feel a little more depressed? Hell yeah. That's different than clinical depression and clinical anxiety. So what I'm reading in this here is that you've got a cohort of people who had a predisposition, right, who were already depressed, who have gotten worse. And that doesn't mean that there aren't additional people out there who are suffering. They can't get in to see a clinician to get the diagnosis.
Yeah.
That's what I'm worried about at this data. But what it's also telling us is that some people are resilient. And good for them, and they'll continue to be resilient. And we know that from other data. So, and I think at some levels that's good news.
Yes.
Yeah, fantastic. Yeah, thanks, Hayley, and thanks, Carl, very much. All right. Hayley, maybe one for you now. Do you have the most common Omicron BA.2 strain?
Yes, we do. We have BA.2 RVPs on our website. We also have the original Omicron BA.1 and BA.1.1. And we currently have 3 new Omicron variants in the pipeline. So we will have 6 in our catalog soon.
All right, so now it's my turn to ask you a question. Crystal ball question. How many more variants are we going to have to deal with here?
No one knows, and no one can predict. With all the data we already have, there's no way of predicting. We had no way of knowing that Omicron would pop up in South Africa, where I'm from, while I was actually there last Thanksgiving.
Wow.
You know, this variant that had the craziest number of mutations that is not related to any other variant around the world except for the original. It's related to the original one that came from Wuhan. There's no way of knowing what is, you know, boiling underneath the surface, what's going to emerge, how well it'll spread, what advantages it'll have over the current strains. You know, when Delta was spreading around the world, we thought that was going to be the most virulent, the most transmissible. And now Omicron has taken over. And not only that, the Omicron we're seeing right now isn't even the first one that came out. It's the second one, BA.2. And there are 3 more siblings that have come out in the past 2 months. It's completely unpredictable. And all we can do is just get good vaccine coverage. That's all we can do, is just, you know, the theories of herd immunity are just not applicable. Because these variants are spreading. Yeah, because you can have, you know, as Liam was saying, you can have both your vaccines and a booster and still catch a virus. It's not that the vaccines aren't working, they're working brilliantly, but you can still get a low-level infection, low-level illness. So all we can do is get good vaccine coverage, wear our masks if we're in close proximity with other people, and wait it out. Just continue to be careful. It's not over.
Yeah, so sometimes you hear over time these strains generally tend to get less severe, maybe more infectious, but usually less deadly over time. And I suppose it sort of, at least superficially, seems like it. It seems like the earlier strains did seem to have higher, you know, mortality rates. Hayley, does it seem like we might be continuing in that trend? It couldn't reverse, could it? Go from Omicron to something that is more deadly?
Well, it's very tricky to know which strains are more virulent, because Omicron is not killing as many people as Delta did, for example, or Alpha and Beta, but so many people in the world are vaccinated now. So that decreases the severity of your symptoms by like 90%. That decreases your chances of severe disease, hospitalization, and death. So we've got a lot of factors at play right now. But yes, in theory, a more virulent strain could come out.
Yeah, definitely. All right, we'll get to the next question in just a second. I think I have to refresh my page to reset the questions. 1 sec.
Can I ask you another question, please, Dr. Marcy, quickly?
Of course.
So you said there's a 2-year waiting list for mental health appointments at your clinic.
Yep.
What can be done? What is needed? Is it more funding? Is it more therapists? What can be done to get those people help? Because 2 years is a long time to not be able to be treated.
Yeah, it's a great question, and I think it's all of the above. I mean, I think the good news is we've seen a massive investment from the venture capital community into telehealth for mental health, which mental health lends itself very well to. I mean, it's not the same as sitting across from someone, but it is one of the fields of medicine where I think telehealth will have huge advantages over time in terms of accessibility. I'll give you another statistic. So prior to the pandemic, our clinic, like many clinics, had a no-show rate of about 20%. That's 1 in 5 people just, the bus broke down, I forgot my appointment, etc. That went down to less than 5 during the pandemic, because I'm going to call you, I'm going to find you, right? Or you got your phone and you're going to get on the Zoom thing, and you have fewer excuses. And for many patients for whom, it's terrifying to leave the house, that's actually great. So I think investing in telehealth and models that scale. I think the other thing we're starting to see is the use of non-licensed sort of paraprofessionals, right? So, how do we train more coaches and patient advocates to help with care navigation, to then also do triaging, so that the patients who really need to see me do, and patients who maybe can be seen by a primary care clinician or a telehealth clinician can get that. And that's really hard to do. But at the end of the day, you're pointing out the reality. And you talk to these telehealth companies who are incredibly well funded. They're all trying to hire the same people. I mean, there are only so many nurse practitioners who are specialized in mental health, and physicians who are psychiatrists. And they're even talking about recruiting family practitioners and primary care docs to help on the mental health side. We just need to increase the training and the incentives to get more people to sign on to the field, which gets to the final one, which is ultimately, reimbursement rates from third-party payers, need to step up and need to be high enough to create the types of incentives that, allow mental health workers to do what needs to be done, which is to think very comprehensively about patients, to communicate with primary care doctors, to think about your social health as well as your mental health, to refer to therapy, because medications aren't the be-all and end-all. And then you take all of this, and it's much worse for young people, because the rates of increase of every category of mental illness for young people is going up faster than even it is in adults. And there definitely aren't enough pediatricians. So that's where now we have to talk about, how do we teach mental health in the classroom, right? How do we teach resiliency and give parents the skills they need, to deal with, kids who are become suicidal at early ages, and it's really scary. So I think we all have to kind of work together. It takes a village, as they say, to make sure that we're thinking about this topic as broadly as we can.
Yeah, that's so true. I know that, Carl, you and I both have children that are similar ages. They look to see how their parents respond in times of stress.
Anxious parents make anxious kids, as they say. So yes, which actually is a great point, which is, as parents, we have to take care of ourselves. Put the oxygen mask on you first, and then your kid. And that's hard to do.
Yes, it is.
That's a good point. Dr. Marcy, I wanted to ask about, you said, that the in-person or the, telehealth, it does seem like a great option for this kind of thing. And it's great to hear that the no-show rates are obviously much lower, but you mentioned that it doesn't quite have the same, it's not quite the same. Are there data that shows that it's, slightly less impactful, or is there any, evidence to support it less, compared to a one-on-one meeting?
Yeah. Well, it's a great question. I think the data is starting to emerge, and I think it's a mixed bag. And what do I mean by that? I think the obvious advantages are convenience, the no-show rate going down, the economics of just having clinicians work from home and not having to have an office with overhead that you'd have to pay for, are very attractive, particularly with the reimbursement rates we talked about, and the like. I think where you lose a little bit is, I learn a lot about watching my patients walk down the hall, right? Are they limping a little bit? Are they moving a little slower? I can smell the alcohol on some of my patients' breath. And I'm like, okay, or, gosh, maybe you're not keeping up with your hygiene. Those are sort of subtle cues you learn as a clinician to watch for, that you just can't do. And then I think, there is a certain intimacy to face-to-face connection. And we know that from other data, whether it's mental health or otherwise, that you just cannot replace the amount of information we get from these nonverbal cues from being with the presence of someone face-to-face. So I think hybrid's going to be the way to go. And then I also worry a bit about, most of my patients are established. So I was seeing them before the pandemic. So I had already had a face-to-face relationship with them. I think it's a little bit trickier, particularly for some patients, to establish rapport and that therapeutic alliance, which is so key to outcomes, through only a computerized and digital connection. I think it can be done, but I think it is going to require some new skills and new training that we probably haven't even imagined yet. I mean, I'll give you a quick vignette, which is a couple months ago, I was talking to a fourth-year resident who was just finishing his training, and this is a couple months ago, so that means a lot of his training was during the pandemic. He has never sat face-to-face with an outpatient. Now, inpatient, he's been obviously, but as an outpatient clinician, he's never actually met a patient face-to-face in his training. And I was just like, wow. Like, what is the impact of that over time? We don't know. We just don't know yet.
Yeah, really interesting. I guess something to definitely, I guess we'll see as the data emerge.
We'll continue to learn. Yeah, we'll continue to learn, and people are already starting to study it as you would expect them to. And I think on balance, it will be positive, is my sense.
Yeah, great to be optimistic. I think in the interest of time, we have time for one more. And Hayley, I think I'll direct it to you. You mentioned that this is an exciting time in your career with all the work that you've done on COVID-19. What do you think are the biggest takeaways from your work that will inform future vaccine and antibody development for potentially even the next world-changing virus?
I think one crucial thing we've learned as scientists is how amazing mRNA vaccines are. This is the first time they've been rolled out globally, and they are incredibly effective and very well tolerated. So mRNA vaccines, this has proven that they are an amazing technology. I think what's also been proven to scientists is how fast we can come up with solutions when we all work together, when there's a lot of funding, a lot of trial participants volunteering, and when we all put our heads together, we can come up with vaccines and antibody treatments really fast. One thing that I hope the pandemic has done for the world population is made them see how effective vaccines are. I think that most people on the planet know someone who's died of COVID. A lot of people have had it by now, and I hope this has proved to people who don't believe in vaccines that they work. And so I hope vaccine acceptance rates have gone up, and that if this happens again, and it will, that we'll be able to curb it, and we won't still be going through these waves of infections 2 years later.
Yeah, definitely. I mean, that's its all, vaccine acceptance and, all the, rhetoric and discussion around that is its own, total can of worms. But I completely agree. I mean, it's pretty heartbreaking to see a lot of people, even in the face of, all this mounting evidence, that don't believe it. And it's so tough, and it's so important for people to really, I think, take a stance, and for, really, the people that have power to, use their platforms to potentially, tout vaccines. It's so important to really send the right messaging to the right people. But yeah, that's a really, really great point. So appreciate that. So I guess, thanks so much, both of you, for the really great discussion in the Q&A and for your great presentations. It's been such a pleasure having you with us.
Thank you so much, Liam.
Yeah, my pleasure. And thanks everyone for joining us today for the presentation. And of course, also a big thanks to Integral Molecular and OM1 for sponsoring this cutting edge conversation. We now invite everybody to take a moment to fill out the survey, which is popping up on the screen there. We really appreciate all your input and any feedback you can provide. If you still have a question, it isn't too late. You can still submit it using that Ask a Question panel, and we will be forwarding it on to the speakers following the event. If you'd like to learn more about Integral Molecular or OM1, you can explore the resources tab there. We've added a few links. And if you're interested in seeing what's coming up next in the Cutting Edge Conversation series, you can click the register button at the bottom of, I believe, on the slide here. Yeah, on the slide there to learn more. So in closing, we hope you enjoyed this Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.